Think about the last time you accidentally cut yourself. Perhaps it was while you were chopping an onion for dinner, or maybe you were shaving and got a little too close. Not me. But in all cases, that injury set in motion a biological process that maintains hemostasis and stops blood loss. That is coagulation. Now, blood coagulation has been studied for a very long time, probably one of the most well-studied ever, because maybe for an obvious reason. Bleeding is bad. But what we've learned over the last several decades of research is that coagulation is a complicated, multi-scale phenomenon. A single length of blood vessel has many interconnected parts. There's biochemical reactions, fluid flow, the biomechanics of blood cells, and a whole host of other important features. And while this system is working correctly, it's easy to acknowledge that complexity without really understanding it. But when something goes wrong, we need to know why. We need to know why. Hemophilia, yes, is just such a case. In bleeding, hemophilia is caused by a lack of certain proteins. But what's fascinating about hemophilia, though, is while two people may have the same protein deficiencies, one may bleed a lot and the other not at all. This means, though, that protein levels alone cannot tell the entire story. We could do experiments, each testing a single variable to see what happens, but experiments are very costly, both in terms of time and money. And the number of variables to look at is huge. My research group thinks there's a better way. We are building a mathematical model, one that includes not only the basic biochemistry, but all these features as well. Working with a mathematical model, honestly, is not too different from doing experiments in a lab. In fact, I really think they're two sides of the same coin. Both start with a simplification of a system and develop something new, either an experiment or a model, and after conducting the experiment or solving the equations, you get new information that goes into reformulating a hypothesis. The advantage of a math model, though, is we can do it very quickly and under very diverse situations. Once this model is finished being built, I am going to work to identify a small class of important parameters. These are the ones that are highly sensitive. And then I'm going to pass information off to an experimentalist whom we are working with to do more extensive research on just that small set, though. In this way, math modeling is really a tool that allows us to use our limited resources much more efficiently. In the end, our goal is to not only learn more about coagulation, but to develop better detection and treatments for hemophilia. Thank you very much.